Welcome to Biomanufacturing and Fermentation Technology. This is your monthly bioprocess and biomanufacturing intelligence, the data, the decisions, and the scale-up realities shaping what comes next. Bioprocess Intelligence Bulletin, 2026 key points we are looking forward in this breakthroughs and manufacturing trends. 1. Continuous perfusion can halve carbon footprint and plastics versus intensified fed batch, moving continuous MAB platforms toward routine use. 2. Hybrid mechanistic VI digital twins from day zero let teams virtually test thousands of flow sheets and cut wet experiments. 3. Precision fermentation is mainstream, adding AI and automation yields about 30% higher efficiency and better economics. 4. Sellers has raised over $600 million to industrialize automated CGT smart factories, now operating at near commercial readiness. 5. Continuous and intensified processing is now a core sustainability strategy, not just a capacity play, with 40–50% environmental gains. 6. Microbial fermentation markets are growing at roughly 6–8% annually, driving both large stainless and flexible modular capacity builds. 7. CGT manufacturing is shifting to automated, single-use, digitally controlled platforms dominated by specialized CMOs via DMOs. 8. CDMOs are in a build-out wave, committing billions to integrated biologics, peptides, and advanced modality manufacturing hubs. 9. FDA, EMA guidance now actively supports continuous manufacturing and lifecycle CMC planning, reducing regulatory uncertainty. 10. Competitive edge is moving to process intelligence, validated digital twins, LCA-T-driven design, and platformized CGT fermentation services.
The true revolution happening in medicine right now, it isn't actually about what we're inventing.
Right.
It's entirely about how we're physically making it. Because if you look at the landscape right now in March 2026, biological manufacturing is just, it's unbelievably messy.
Oh, it really is. It's so unpredictable.
Yeah, and wasteful, like it's actively bottlenecking human health. So today, we are doing a deep dive into the intelligence on how the industry is finally fixing the factory. I'm your host.
And I'm your expert for today, bringing you a really fascinating stack of bioprocess and biomanufacturing intelligence.
Exactly.
Right? Because, honestly, the factory it's the single biggest hurdle between a miraculous scientific discovery and that discovery actually reaching a patient.
Which is crazy to think about.
It is. We're going to map out this complete overhaul of the physical infrastructure of biology. So we're looking at, the transition from these massive wasteful vats to continuous processing, the leap into process digital twins, and then finally, the deployment of these full robotic smart factories for living human cells.
I really want to trace the logic of why you, listening to this deep dive right now, should care about the plumbing of a biotech facility.
Yeah, it sounds a bit dry on the surface, but it's not.
Not at all. Because whether you're like an investor looking at synthetic biology or a patient waiting on a specialized therapy or just someone who tracks global sustainability, the harsh reality is that the discovery phase is just no longer our primary limitation.
Right. We have the molecule.
We understand the genetics. The barrier is that biology actively resists being put on an assembly line.
Biology is inherently chaotic. I mean, when you try to force a living cell to act like a microchip factory, the cell rebels.
It really does.
And historically, the industry's response to that chaos has just been brute force. Which brings us to the first major shift we need to examine today.
The end of cook and dump.
Exactly. The entire sector is desperately trying to escape this legacy manufacturing method known as fed batch processing. And they're moving towards something called continuous perfusion.
I had to look really closely at the mechanics of fed batch because, on the surface it sounds simple, but the scale of the waste is just staggering.
It's basically a giant, incredibly expensive pot of soup.
Yes, that's a perfect analogy. In a fed batch process, you basically fill a massive stainless steel vat. I mean, sometimes holding 10 or 20,000 liters with a liquid nutrient broth and your engineered cells.
You feed them, and you just wait.
Right. You let them simmer in this giant pot for weeks, hoping they produce your target molecule, like a monoclonal antibody, for instance.
And monoclonal antibodies are incredibly complex. These are those Y-shaped proteins we use to treat everything from arthritis to cancer.
They're super delicate.
Very delicate. And in a fed batch system, as those cells spend weeks multiplying and secreting the antibodies, they're also secreting toxic byproducts.
Like ammonia and lactate.
Exactly.
Right.
So by the end of the run, those expensive, highly engineered cells are essentially swimming in and being poisoned by their own waste.
Which means the cell viability just drops off a cliff. And the worst part is you don't really know your final yield or the exact quality of the proteins until you stop the entire process and harvest the whole multi-million-dollar batch all at once.
You're flying blind for weeks.
And if like a stray bacteria got in or the temperature fluctuated and the proteins folded incorrectly, you're literally dumping millions of dollars and weeks of labor straight down the drain. It's like making that massive pot of soup and realizing at the very end you used sugar instead of salt.
And the whole thing goes in the trash.
Yeah.
The environmental and financial cost of that failure rate is just completely unsustainable.
Yeah.
Which is why there's this UK-led initiative, it's called InspireMed, driven by Fraunhofer UK. They are aggressively pushing the industry toward semi-continuous perfusion for these biologics.
Okay, let's break down the actual mechanism of continuous perfusion then, because it radically changes the environment for the cell, right?
It does. It completely changes the game.
Yeah. Instead of sealing the cells in a giant vat to slowly choke on their own waste, perfusion is dynamic. It's like a smart conveyor belt. You're constantly pumping fresh nutrient media into the bioreactor,
while simultaneously filtering out the toxic byproducts.
and extracting the finished, purified antibodies at the same time.
Yeah, you're basically creating an artificial circulatory system for the bioreactor. The cells are kept in this state of constant health because their environment is pristine,
Right.
so they survive longer, and they produce significantly more product per liter of space. And the footprint reduction is just phenomenal.
I saw the numbers on that in the intelligence, the Sartorius models.
Yes. Sartorius did these early life cycle models, and they calculated that moving to an end-to-end continuous process for monoclonal antibodies can cut a facility's carbon footprint by up to 51%.
That is massive. And they also model, I think it was up to a 57% reduction in plastic waste.
Yep, 57%.
Because you don't need thousands of single-use storage bags to hold all those massive batches of media anymore. Plus, a 54% drop in overall CO2 emissions compared to a standard intensified fed batch facility running at, 2,000 kilogram annual throughput.
And those emissions drops come from the fact that continuous facilities are just physically much smaller.
Because you're producing more with less space.
Right. You don't need the massive HVAC systems to maintain giant clean rooms, and you don't need these colossal water purification systems for buffer prep. Contract manufacturers are already seeing sustainability improvements in the 40 to 50% range just by adopting this.
Okay, but here is where I have to push back a little on the timeline.
Sure.
Because if a continuous smart conveyor belt system cuts carbon and plastic in half, keeps cells happier, and stabilizes the cost per gram, which, by the way, is vital for getting cheaper biosimilar drugs to market.
Absolutely.
Then why is the intelligence rating this at a technology readiness level of only 6 or 7? Like, we have the pumps, we have the filters. What's the holdup? Why isn't everyone doing this right now?
It's a fair question. The mechanical unit operations, they work perfectly. The holdup is entirely the regulatory reality of proving control.
Proving control to the FDA.
Yes. When you're continuously extracting a drug that will be injected into a human being, you cannot wait until the end of the month to test if it's safe. You need something called PAT.
Which is process analytical technology, right?
Exactly.
Meaning you have these inline sensors that are testing the chemical composition of the drug continuously as it flows through the pipes.
Yes, and regulators like the FDA require an overwhelming burden of proof that your sensors won't drift or fail over a multi-month continuous run.
Ah, right.
The FDA has their Q13 guidance, which supports continuous manufacturing in theory, but in practice, they demand multi-product, multi-year validation data to prove your entire control stack works.
Which creates this massive catch-22.
It really does.
Because you can't get regulatory approval without years of data, but you can't afford to build a $500 million continuous physical plant just to gather test data on a drug that honestly might fail clinical trials anyway.
Exactly. And that bottleneck forces a complete rethinking of how we engineer these facilities. If you cannot afford to build the physical plant to find out if your process works.
You have to bypass the physical world entirely.
Precisely. Which takes us straight into the second major evolution. The process digital twin.
This part is wild. We're looking at a platform from a company called New Wave Biotech, and they couple mechanistic models with AI to screen thousands of manufacturing routes in silico.
Right. And to understand the power of a hybrid mechanistic AI model, we really have to look at what happens when you skip it.
The classic downstream surprise.
The classic downstream surprise. It's the absolute nightmare in biomanufacturing. A team designs a brilliant process in a tiny 2-liter glass jug in a laboratory. It works flawlessly.
Everything looks great on a small scale.
Exactly. So the company spends $400 million building a 10,000-liter commercial facility based purely on that lab data.
But fluid dynamics do not scale linearly. Like a 2-liter jug behaves very, very differently than a massive 2-story steel tank.
Drastically differently. Inside a commercial bioreactor, you need these massive metal impellers, basically giant mixing blades, to ensure oxygen reaches all the cells.
Right.
But if those blades spin too fast, the sheer stress literally physically tears the delicate cell walls of the microbes apart.
Oh, wow.
But if they spin too slow, the cells at the bottom of the tank suffocate.
So the mechanistic model isn't just a spreadsheet, it's like a flight simulator.
That's a way to put it.
You want to crash a virtual plane 1,000 times in software, simulating the exact physics, the fluid dynamics, the shear stress, the heat transfer, so that when you build the real one, it flies perfectly on the first try.
Exactly. You're stress testing the physics virtually.
Right.
You find out that your cells produce the drug so fast that your purification filters will clog.
Right, line rate limitations.
Or you find out your facility's utility system literally cannot pump enough chill cooling water to keep the massive bioreactors from overheating. You discover all of this in software where fixing it costs absolutely nothing.
I was tracing the logic on the economic impact of this, though, because I have a question. The intelligence notes that these digital twins are delivering efficiency gains of around 20 to 30%.
Yep.
Now, on the surface, if I upgrade the software on my laptop and it runs 20% faster, I mean, that's nice, but it's not exactly world-changing.
Sure.
Are we guilty of overhyping the AI here, or is a 20% gain actually a massive deal in this industry?
I get why you'd ask that, but no, it's not overhyped. In this context, a 20% yield boost is literally the difference between life and death for a commercial drug program.
Really? Just 20%?
Think about the capital expenditure. If your process digital twin optimizes your flow sheet so effectively that you get 30% more purified drug out of your existing facility,
Oh, I see.
you might completely eliminate the need to build a second $500 million manufacturing campus.
Yeah.
It's sitting at TRL 5 to 6 right now, already being used for early development programs.
Wow, okay. So moving those expensive trade-offs upstream, day zero, is brilliant. But once you've optimized the factory floor virtually, you still have to deal with the actual workers on the assembly line.
Which brings us to the microbes.
Yes. In biomanufacturing, the workers are the microbes. And precision microbial fermentation is transitioning from like a niche scientific endeavor into the default manufacturing platform for the entire global bioeconomy.
The scale is hard to comprehend. The market for microbial fermentation in pharma alone is projected to hit $27 billion in 2026.
$27 billion? That's insane.
And we're talking about relying on engineered yeast and bacteria to produce highly targeted enzymes, complex amino acids, and high-value therapeutics.
And the reason that market is exploding is because of how precise we've become with metabolic pathway editing. Like, scientists can now use tools like CRISPR to snip a specific genetic sequence out of a human or a plant and paste it directly into the DNA of a microbe.
You are fundamentally rewriting the microbe's internal operating system.
Exactly. You're treating them like programmable 3D printers. Instead of consuming sugar and sweating out alcohol like traditional yeast does.
You trick its metabolism into sweating out a breast cancer drug.
Or a rare flavor molecule that normally requires harvesting thousands of acres of plants. You feed them sugar, and they print out specialty chemicals.
And because we're now pairing that advanced strain engineering with the AI-enabled bioreactors we just talked about, the efficiency is spiking.
Yeah, the bulletin mentioned that AI-monitored feed control and oxygen transfer has boosted production efficiency by about 30%, while drastically cutting operating costs.
Which is exactly the threshold required to move this technology out of just ultra-expensive pharmaceuticals and into the food and specialty chemical markets.
Companies like Ginkgo Bioworks, Amyris, Perfect Day, and LanzaTech.
Exactly. They are turning precision fermentation into a multi-billion-dollar commercial reality. The platform itself is operating at TRL 8 or 9.
Okay, but again, I've got a pushback based on a sharp caveat in the bulletin.
Let's hear it.
It warns that stream breakthroughs alone rarely close the business case without equal innovation in recovery and purification. So we can engineer a brilliant yeast that pumps out a valuable protein, but we're still struggling to actually get that protein out of the vat.
It is the ultimate bottleneck.
So are we basically building a Ferrari engine, these incredibly fast, genetically optimized microbes, but forgetting to install the brakes in the transmission?
That is a very accurate way to look at it. You have a vat containing billions of yeast cells, nutrient broth, cellular debris, and your target protein.
It's just a biological soup.
Exactly. And separating that single fragile protein from the soup without destroying it requires massive, highly specialized downstream purification lines.
Centrifuges, chromatography columns, specialized filtration.
Yes, and because that downstream infrastructure is so incredibly capital intensive, it's forcing a massive structural shift in the business landscape. We are seeing a hard split between the therapy innovators and the manufacturing platform owners.
Right. Because the synthetic biology firms, they're doing the brilliant genetic engineering to design the strains.
But they simply do not have the billions of dollars required to build the hardened facilities to purify the product at scale.
Facilities that have cleared the massive hurdle of GMP good manufacturing practice certification from the FDA.
Precisely. So the innovators are forced to turn to massive contract development and manufacturing organizations.
CDMOs.
Right, CDMOs. Companies that do nothing but build and operate these GMP-ready campuses. The innovators hold the intellectual property of the microbe. The CDMOs hold the physical means of production. You really need both to survive.
Okay, so programming a microbe to secrete a protein is an astonishing feat of engineering. But it still relies on extracting a byproduct. The absolute limit of our manufacturing capability, like the highest stakes environment in biology, is when the final product isn't a secreted protein at all.
Right. It's when the product is a living, breathing human cell.
Cell and gene therapies, CGT. This requires an entirely different paradigm of manufacturing because you are not just growing cells in a vat.
Not at all. I know you were looking at the intelligence on Cellar.
Nice, sellers. They've pulled in over $612 million in total funding, including a recent $257 million Series D.
A massive amount of capital.
Huge. And their entire mission is to build what they call an IDMO, an Integrated Development and Manufacturing Organization. They are building automated, enclosed, smart factories specifically for cell therapies.
And that volume of capital makes total sense when you look at how fragile current cell therapy manufacturing is.
It's so manual.
It really is. Take an autologous cell therapy, for example. You extract T cells from a specific cancer patient, you transport them to a facility, genetically engineer those specific cells to recognize and attack the patient's tumor, multiply them, and put them back into the patient.
It's brilliant science, but as a manufacturing process, it's terrifying. The analogy I kept thinking of is that it's like bespoke artisanal tailoring. Each dose is hand-stitched by a master craftsman.
That's exactly what it is. It's like running a volatile, high-end restaurant kitchen where the ingredients are alive. They change shape depending on the room temperature.
And if you cross-contaminate one patient's cells with another's, it's a fatal disaster.
Right. You have highly trained scientists in cleanroom suits, manually pipetting fluids and carrying bags of living cells between incubators.
Which is why Cellares is trying to introduce the automated Henry Ford assembly line to living cells.
Exactly. That intense manual labor, combined with the extreme risk of human error, is why operator variability is the main driver of costs in CGT.
We're talking about therapies that cost hundreds of thousands of dollars per dose.
Yes. And Sellers is attempting to take that chaotic, artisanal clean room and turn it into a robotic vending machine. They're deploying modular, fully enclosed robotic systems.
The machine handles everything from the initial cell processing all the way through to what the industry calls fill finish,
which is the incredibly delicate final step of getting the living engineered cells into the sterile IV bag or syringe without killing them.
Right. But there is a massive paradox buried in the report about this transition.
The talent shortage.
Yes. The data warns that even with all these robotic smart factories, physical capacity expansions risk being underutilized unless the talent pipeline catches up. And my question is, if we are automating everything with robots and AI, why are we worried about a talent shortage?
Because the nature of the labor changes fundamentally. You no longer need the manual pipetting skills. What you desperately need are highly specialized operators who deeply understand data analytics, process digital twins, and AI control systems.
Oh, I see. So the robots handle the physical risk, but you need an operator who knows how to interpret the telemetry coming off the robot.
Exactly. And that specific intersection of skills, a deep understanding of living cell biology paired with advanced systems engineering, is incredibly rare.
Yeah, it makes sense.
The value is shifting entirely toward those who can manage highly automated network facilities. The next 24 months will be the real proving ground. These automated CGT systems are sitting at a TRL of 7 or 8. They're deployed in the field.
Now they just have to prove they can run multi-product commercial campaigns without deviation.
Right.
When you pull back and look at the entire landscape we've covered today, the through line is incredibly clear. We are really witnessing the end of brute force biology.
We absolutely are.
I mean, we explored the pivot from wasteful fed batch vats to highly precise, continuous perfusion, filtering out toxic waste in real time.
Right.
We examined how mechanistic AI models are stress testing the fluid dynamics of virtual factories before a single drop of concrete is poured.
Yep.
We looked at microbes being genetically reprogrammed to print therapeutics, and finally, the push to turn chaotic clean rooms into robotic smart factories for human cells. The era of biological process intelligence has officially arrived.
It really represents the maturation of an entire scientific field. We're finally applying industrial process engineering to the unpredictability of nature. And, for you listening, the stakes for getting this right extend far beyond corporate balance sheets.
The stakes are the actual accessibility of the future.
Exactly. These manufacturing bottlenecks dictate whether a life-saving personalized genetic therapy remains a $2 million luxury for a tiny fraction of the population or becomes a standard, widely accessible treatment.
And whether our medical supply chains are sustainable for the planet.
Right. Whether they continue to be a massive source of carbon emissions and plastic waste, or if they transition into highly efficient, sustainable loops.
It's the invisible infrastructure that will define the next century of health. But there's a final lingering implication to all of this that is worth turning over in your mind after you finish this deep dive.
Ooh.
We are rapidly moving toward an ecosystem where the manufacturing platform owners, the massive CDMOs, and the companies operating these proprietary AI-driven robotic smart factories, they are going to hold all the physical leverage.
They own the hardened facilities. They own the complex regulatory validation data.
Right. They control the means of biological production. Which raises the question, as the complexity of the factory continues to outpace the complexity of the discovery, will the biotech companies that actually invent the miraculous new drugs eventually just become IP licensing firms?
Wow. That's a shift.
Will the manufacturing platforms quietly end up controlling the power dynamics of the entire global healthcare system? It is definitely worth keeping a very close eye on who actually owns the factory floor. Because the scientist holding the glowing vial might get all the headlines, but the engineers building the smart factory are the ones quietly taking ownership of the future.
That's this month's Bioprocess Intelligence. If you're turning biology into manufacturing at scale, these are the signals to watch. I'm Prasad Ernala. See you next month on Biomanufacturing and Fermentation Technology.